Hello and welcome back to Bio 2. This is a lecture on chapter 2A and 2B. Go ahead and get out your lecture notes and fill them in as we go. Remember to pause as you need and take plenty of notes. I'm going to share my screen here. Okay, here we go. Chapter 2A, it's all about atoms, molecules, and pH. Okay, what are some basic concepts and terminology that you will need to understand for this chapter? Well, first, there are three states of matter: solid, liquid, and gas. There's actually a fourth state called plasma, but we're not going to worry about that. It's found only in the sun and in some scientific laboratories. Solids, they maintain their shape and volume under normal circumstances. That means they can't be compressed. Liquid, the molecules move past each other, also incompressible. Shape is dependent on the container and the volume is fixed. So for example, if a cup is half full of water, it will take up the exact shape of the bottom of the cup, only on the bottom half if you are on a planet and not out in space. So please remember, liquids are not compressible. The molecules are attracted to each other, not as much as in a solid. The gases are very different than others, though, because the molecules move at a higher speed, running into the walls, generating pressure. There's a very low attraction for each other, and the volume and shape are variable. They always take up the entire space inside of whatever container they're in. These are the three states. What are some more basic concepts and terminology? What is an element? Well, it's an atom or a group of atoms that are all the same. For example, if you have pure gold, 24 karat gold, it's all gold atoms, and that makes it an element. Same with oxygen. If it's pure oxygen, that's an element because it's just oxygen atoms. If a material is composed of two or more different atoms, then it is a compound. So for example, water, while the ancients used to think that it was an element because they couldn't break it down, we know that it can be broken down into both hydrogen and oxygen. So it is not an element, it is a compound. So is water an element or a compound? It's a compound because it's made of two different kinds of atoms, not just one. Now, to see the characteristics of the different atoms that exist on our planet in the universe, there's a thing called the periodic table of elements. I'll be teaching you a little bit about it, but I will not be asking you to memorize the whole thing. It's an arrangement of elements that lists the elements based on their similar properties. And I'll be showing you what that means here in a moment. Okay, this is one example of the periodic table of elements. Now, in this case, for what we're going to be looking at, the colors don't have meaning. So please try to reject the fact that they have different colors. What's important is the columns. Now, notice the top of each of these columns are different Roman numerals. If you're not familiar with how Roman numerals work, each I or line here is a number. So this is 3. So if you count those 3, and then in Roman numerals, a 1 before a V, which is 5, means take away 1 from 5. So that's 4. There's 5. There's 6, there's 7, and here's 0. Now, in reality, this should say 8, but I'll talk about that more later on. The key here is each of the atoms in each of these columns have similar characteristics, and I'll tell you what those are. The key is understanding that each of the columns is very specific for the kinds of atoms in it. Okay, so let's talk about atoms. Each element consists of only one kind of atom. There can be lots of those atoms, but it has to be just one kind. An atom is the smallest unit of matter that still retains the properties of the element. Now, please note that that does not mean the atom is the smallest unit of matter. But when you look at the subparts of an atom, those subparts are equal amongst all different kinds of atoms and elements. So if you don't have at least a whole atom, you don't have that material that you're talking about. For example, gold. You have to have all those particles and all the right configurations for it to be gold. When you're looking at the individual particles, they're the same across all atoms. Now, if you look at this figure here, it shows two different ways that you can show an atom. One has discrete rings showing the electrons. Notice here, electrons have a minus charge. They go around the outside in shells. Protons and neutrons are in the center. Notice protons have a positive charge. Neutrons have no charge. So try to remember this. Protons positives, electrons negative, and the way to remember that is there's a negative symbol on the end of a battery. We'll be talking about more later, and that's where the electrons come out. Neutral is neutron. So try to remember neutral and neutron, electrons are minus. Okay, let's talk a little bit more about atomic structure. The electrons in an atom are arranged into energy levels or shells. The behavior of atoms is due to the number of electrons in their outermost or valence shell. Please notice it has to say valence. The word valiant means brave, and it's not the same word, even though it sounds similar. So please be careful when you're studying for this, not to pick answers on my test that sound similar, but are either exact or equivalent. So valence means outermost shell. Stable atoms are those that have 8 electrons in their outer or valence shell. On the previous image that I showed you, the periodic table, all the ones in the far right column are called the noble gases. Why are they called noble gases? Because as in the nobility, they don't want to mix with the common folk or other atoms. So only those in the far right-hand column don't react, don't mix with other elements. When atoms that do not have 8 electrons, all those other than the noble gases, when they don't have 8, they seek to fill their outer shells until they usually have 8. So the idea is that they want 8. If they're very small, they want 2. But they seek to fill them by forming bonds. Now, notice hydrogen here only has 1 ring around it. The innermost ring of all atoms has only space for 2 electrons. Now, when you go to the next layer, the next outer shell, there's room for 8. Let's go back to hydrogen. Notice that there's 1 there, 1 negative electron, and an empty spot. So it's electrically neutral because there's 1 negative and 1 positive. There's no neutrons here. But if we look at carbon, you see that the innermost shell is filled with 2, but the outermost shell only has 4 with 4 empty spots. What does that mean? It means that carbon is going to try to gain 4 more to get to 8. It's going to do that by forming bonds. Notice here nitrogen only has 3 empty spots, so it's going to form 3 bonds because it wants to get up to 8. And oxygen only has 2 empty spots, so it's going to form 2 bonds. Okay, molecules and chemical formulas. What is a molecule? Well, when 2 or more atoms are bonded together by sharing electrons or by having negative charges, it's called a molecule. A good example of that is water, H2O. H is the symbol for hydrogen. In the same way that we use an S with 2 lines in it to be the symbol for dollars, we use H as the symbol for hydrogen. Now, there's a big difference between the name and the symbol. The symbol is just 1 letter. The name is the full word, water, or hydrogen in this case. The subscript 2 means that there are 2 of them in the molecule. Now, in math, if it was x squared, the 2 would be up above. But because it's down below, it means that there are 2 hydrogens and 1 oxygen. When there's nothing written after the letter, it means there's 1. The O is the symbol for oxygen, and since there's no number after the O, you can assume that there is only 1. If there were none in there, it wouldn't be written. So think of it kind of like math, but instead of the numbers being up above, they're down below. Okay, trends in the periodic table. What is the happy family? Well, I told you the atoms that don't have 8 already are seeking to get the rest of the electrons they need to get to 8. Most atoms, as they sit on the table, other than the noble gases, are unhappy. What do we mean by that? They're unstable because they don't have 8 in their outer shell. So they can't sit like that. They have to find more electrons. Atoms can gain electrons by forming bonds with other atoms. This is the main way that they do it. A bond is formed when 2 atoms share 2 or more electrons between them. Please note, a bond is 2 or more electrons. They always come in pairs. So 1 bond is 2 electrons being shared. 2 bonds is 4 electrons. So every time there's a bond, you multiply that by 2. That's how many electrons are being shared. You can determine how many bonds an atom will usually make by looking at its family number. How do you do that? Well, you have to take the number 8, subtract the family number, and that's the number of bonds it will form. Since carbon is in family 4, it only has 4. So you take the number 8, subtract 4, giving you 4 bonds. So by forming 4 bonds, it gains 4 electrons and becomes happy. So when they share electrons, they both get to count them as electrons around them, and that makes them happy. So notice here, this column only has 3 on the outside. This number here has how many it already has. This is not the number of bonds it will form. So all these in here seek to form 5 bonds because 5 plus the 3 already here equal 8. In this column, carbon, silicon, germanium, There's 4, so it seeks to form 4. Nitrogen already has 5, the V is 5. So it wants to form 3 more. 6 forms 2 more. Here, it's got 7, forms 1 more. And then over here, they already have 8, except for helium, which has 2. So they don't seek to form any. So this really should say 8 on top. It says 0. This is the number of bonds that are going to be formed here. The rest of this, you have to take 8 minus the family number. That's the number of bonds it will form. How many bonds will nitrogen form? Since it's in family 5, forms 3. How many bonds will boron form? There it is. It's in 3, wants 5 more. That means 5 bonds. I hope that helps. Okay, 25 elements are essential to life. 4 of these, O, C, N, and H, make up about 96% of the weight of the human body. Now, my students often say, Mr. Sage, I thought we were made up of water. It's true, but look at O and H make up water. So if you take away most of the water, you've taken away the O and the H. Notice how much there is in living things, mostly oxygen. Well, how's that possible if there's twice as much hydrogen as oxygen in water? It's because oxygen is so much bigger than hydrogen. Hydrogen is a very small atom. Trace elements occur in smaller amounts, and you can see down here all of these other elements that we have in us in smaller amounts. Okay, what is an ion? Atoms and molecules are electrically neutral and they have equal number of protons and electrons, and that's how they are sitting on the periodic table for the most part. When an atom or molecule gains or loses an electron or 2, it's called an ion. So if it gives up 1 or picks up 1, it becomes an ion. If an ion has a positive charge, which means it's lost an electron, it's called a cation. So remember, if you lose your negativity, you become positive. So for example, because a hydrogen ion has a plus symbol, that means it's lost 1 of its electrons, and only add 1 anyway. So really, a hydrogen ion is just a proton, but the positive charge means it's lost an electron. If an ion has a negative charge or gained an electron, it's called an anion. It's not an anion, it's an anion. It's like 2 words put together, like cat ion, anion. Look at the word ion in there, that'll help you. So for example, the hydroxide ion has picked up an electron. Now, what happens when you mix these 2 ions together? The extra electron here and the missing one here work together, and this forms water. So when you have hydrogen ions and hydroxide ions, they mix together to make water. Okay, talking a little bit about ionic bonds to start. Now look at sodium up here, this purple atom. It only has 1 electron in its outer shell. So it could form 7 bonds, but that's a hassle, and there's not room for 7 things to attach. So what it does is it cheats. Instead of adding 7 more, it donates its outermost electron to an atom such as chlorine, so that now, look at sodium, it has 8 around the outside. But because it's given away 1 of its electrons, it has a positive charge. The chlorine could bind with something, but it could just as easily pick up a free electron from sodium, filling its outermost shell. Notice it now has a negative charge because it added a negative electron. Now notice this chlorine has a minus charge and this sodium has a plus charge. These 2 become attracted to each other, just like magnets, and they stick together, forming a crystal when they're outside of liquid water. And that's how we have sodium chloride or table salt crystals. When certain atoms are arranged together, one atom with too many electrons, in this case sodium, will donate an electron to another atom that has too few, in this case chlorine. This forms a cation, which is the sodium, and an anion, which is the chlorine. And the cation and anion are near each other, and they're attracted due to opposite charges. And this happens in crystal form when they are when water has evaporated. That's why you find salt in crystals. The attraction between them is called an ionic bond, and it's not very strong, but it does hold them together in the salt crystals. They can be easily broken up. As a matter of fact, sodium and chlorine would much rather be sticking to water. That's why salt dissolves so well in water. We'll be talking more about that later. Covalent bonds. Now remember, we said the valence electrons were the outermost. So covalent means sharing the outer electrons. Let's take a look at the picture before we do the notes. Notice these 2 hydrogens are overlapping their outer shells. This hydrogen brought this one, and this hydrogen brought this one. So now the 2 here being shared can be counted for both. So hydrogen wants 2, this hydrogen wants 2. Since they both brought 1, they can both count them. To show this single bond of 2 electrons, you put a dash here. And that's what we're going to be doing in lab. Notice with oxygen, it's going to be sharing 4 electrons, forming 2 bonds, and that's shown as an equal sign here. sharing 2 electrons. Since these are shared, they can be counted for both. Methane, something we're going to be doing in lab, has one carbon, 4 hydrogens. Each of the hydrogens is sharing 1. The carbon is sharing 4 between them, and it's drawn like this. Water is an interesting one. I call it the Mickey Mouse molecule. Everything wants to be water, and I'll tell you why that's the case later on. But notice that the 2 hydrogens are not equal on the sides. It's because these electrons here are pushing them out of the way, giving it that Mickey Mouse shape. And it's drawn like this, like a V. Let's do the notes. When 2 atoms share 2 or more electrons, this is called a covalent bond, because co means together, valent as in valence shells. You're going to overlap your valence shells, make them stick together. These are powerful bonds normally and difficult to break. But chemical reactions can break these bonds, and I'll show you how this works. What is a chemical reaction? Well, it's the change in the arrangement of atoms and molecules due to rearrangements of the bonds between atoms. Let's take a look at the example down at the bottom. Here's hydrogen gas, 2 hydrogens stuck together by 2 electrons. We saw this earlier. So we have 2 of those and 1 oxygen gas. Notice that the oxygens are together and the hydrogens are together. When you mix hydrogen gas and oxygen gas together and spark it, you get water. Because as I said, most molecules want to be water because water is super duper stable. So notice all of the material is the same on the right, but the bonds are changed. So instead of oxygen being hooked to oxygen, it's hooked to hydrogen, and this reverse for hydrogen. The number of atoms of oxygen and hydrogen on the left side of the arrow is the same as that on the right. Count that up. 2 oxygen, 4 hydrogen. The arrangement has changed. The bonds have changed, and that's what happened during a chemical reaction. The sticks between the atoms represent the covalent bonds in these molecules. So this stick means 2 electrons are being shared. The double stick means 4 electrons are being shared. The materials on the left side of the arrow are called reactants. Why? Because they're going to react. The material on the right side of the arrow are called the products. Now this is different than math. In math, you would say 1 + 2 = 3. In that case, there's no time component. You could easily say 3 = 1 + 2. But in a chemical reaction, the stuff on the left side of the arrow means before. The arrow means something is happening, a chemical reaction. And the stuff on the right is what you get afterwards, the product. So the reactant's going to react, forming products. Now, if there's an arrow on the left side as well, it means it can go either direction. But if it's just one-headed arrow, it always goes from these 2 things to this material here. What are some signs that a chemical reaction has occurred? Well, we're going to see some of this in lab. The color changes to a brand new color. So for example, if you mix a clear liquid with an orange powder and you get a red material, that's a brand new color. But when you mix blue and yellow together and you get green, that is not a new color because that's the color that forms when you blend 2 colors together. So if it's a blended color, for example, white and red making pink, well, pink isn't a brand new color, that's a blend. So if you find a brand new color that has nothing to do with the blended original colors, that shows you that there's been a chemical reaction. When the temperature changes, the 2 chemicals, when mixed together, get hotter or colder. So for example, you may have heard of a cold pack. You squeeze it and break the barrier between them, mix the chemicals together, they get cold. And sometimes they would get hot if it's a hot pack. Same with epoxy glue. I've used epoxy glue many times. You mix the 2 materials together and it gets hot. That shows you a chemical reaction is happening. So when it gets hot or cold, that tells you a chemical reaction is normally happening. When gas is formed, bubbles or foam or smoke usually are seen. Now, when you open a bottle of soda and the bubbles come up, that is not a chemical reaction. That is just gas coming out of saturated liquid. So you haven't mixed 2 chemicals together, you just release the pressure on the soda, allowing the bubbles to form. But if you mix 2 chemicals together and bubbles form, that's usually a chemical reaction, smoke, gas, etc. When a precipitate solid is formed, only when you mix 2 liquids, a solid material will settle out. This one's different than the others because it only happens when you mix 2 liquids together, forming solid chunks in it. So please make a note that it's only going to happen when you mix 2 liquids together, forming a solid material, and that solid material will settle to the bottom. Why? The word precipitate happens. You guys have heard of precipitation. That's when moisture in the air that's floating in the clouds falls to earth in the form of water or snow. So it's called precipitation because it's now falling out of suspension to the ground. Light formation. For example, fireflies mix chemicals together, deep sea fish, and glow sticks. In a glow stick, you bend it to crack the glass in between the two sides, shake it up. When those two chemicals mix together, light forms, and that tells you that the chemical reactions happen. Okay, so if I ask you which of these 4 or 5 is found in a fire and which one is not, let's take a look at them. Does the color change? Well, let's say we start with a piece of white paper. Does it go to gray or black? Yes, it does. So check for 1. Does the temperature change? Does it get hot? Yes, fires generate heat, telling you that there has been a chemical reaction. What are the two chemicals mixing together? It's oxygen and the paper. Is gas formed? Is there bubbles or smoke or foam? Yes, you do see smoke. That tells you chemical reaction. Has a precipitate form? No, because precipitate only forms when you mix two liquids together. Fire is not when you mix two liquids together. So that's not true. Does light form? Yes, fires do generate light. So 1, 2, 3, and 5 are all evidence that a fire is a chemical reaction. Let's talk about water. Without water, no life can exist on Earth. There may be other planets that can have life with no water. All life on this planet requires water. Water is 1 part oxygen, 2 parts hydrogen, with single covalent bonds between them at an angle. I showed you that earlier. One pole or end has a slight positive charge and the other has a slight negative charge. So remember, in ionic bonds, it was a positive and negative charge. But in this case, it's not going to be full positive, full negative. It's only going to be slightly positive or slightly negative. This is due to the fact that oxygen molecules are so much heavier than are the hydrogens and pull more of the electrons near them. So because oxygen is so much bigger, the electrons are going to kind of stay around the oxygen more often than they are the hydrogen, hence giving it a slightly negative charge because electrons are negative. Because the hydrogen doesn't get the electron as much, it generates a slightly positive charge. This polarity, having a positive end and a negative end, slight, causes the water molecules to be attracted to each other like the ends of a magnet, kind of like what we saw in sodium and chlorine, but not quite as strong. The resulting attraction is called a hydrogen bond. So when you have water molecules near each other, they will tend to be attracted to each other because the hydrogens have a positive charge, slight, and the oxygens have a slight negative charge. These hydrogen bonds are responsible for water's behavior. So I may be asking you to tell me about these hydrogen bonds and how they work, what they are, and how they affect water's behavior on an essay coming up on your first test. Okay, let's take a look at it. There's the oxygen. Notice how big and heavy it is. So the electrons should move towards the oxygen as shown by these yellow arrows. Notice because it has more electrons more often around it, it has this slight negative charge. The hydrogens, having the electrons pulled away from them more often, have a slight positive charge. So notice this top end has a slight positive charge, and this oxygen end has a negative charge. When you put them near each other, the hydrogens from one water molecule are going to be attracted to the oxygens of an adjacent one. And this is going to be true for all the water molecules around them. And this is what holds water together and gives it much of its properties. Now notice that the lines are dashed. These are dashed because they're easily broken. And this is handy because when you jump into water, you don't want these molecules holding together rigidly because you'd break your legs. The water molecules will move past each other, getting out of the way, allowing you to fall into the water without injury. Now we are going to see that these become rigid when the water freezes. You don't want to jump into a frozen pond. But in a liquid water pond, these hydrogen bonds are somewhat flexible. Let's talk about the life-supporting properties of water. Adhesion and cohesion. Well, if you think of the word adhesive means to stick to a surface. Adhesive tape sticks to a surface. Cohesion, when you say a group is really cohesive, that means they stick to each other well. Water sticks to itself, cohesion, and to surfaces, adhesion. This allows water to move up into trees and gives water a surface tension. Now, you could experiment with this by dipping a corner of a paper towel into water. The water will move up into the paper towel because of this adhesion and cohesion, because the water molecules are more attracted to those paper molecules than gravity is pulling them. High heat capacity. Water absorbs heat from warm air and releases it to cool air. So what does this mean? Well, this is how sweating works and how evaporative coolers work and why it is cooler in the summer at the beach than it is in the desert. Why is this the case? Well, it takes a lot of heat energy to make water evaporate. So what happens is when you're sweating, if the wind's blowing across you, it's pulling the high heat energy water molecules away, leaving behind less energetic water molecules, which is cold. So that's what cools your body, because those warm water molecules are being pulled off of you. The remaining water on your skin is cooler, and that's what cools you off. The blood from your body goes into your skin, is cooled by that remaining cool water on the surface, and then is transmitted back to your muscles to cool you off. And this is the same thing that happens in an evaporative or swamp cooler. If you force evaporation, leaves behind cool water, the air moving across the cool water is cooled, and that blows into your house, cooling you off. So large bodies of water warm slowly, and this is why the beach areas are cooler than the deserts in the summer. Because if you have a big body of cool water, the air above it is going to cool off. Water takes heat and energy away when it evaporates. This allows sweating and swamp coolers to work, as I already explained to you. Ice floats due to the fact that water molecules spread apart when they freeze. This is why we have to put antifreeze in old-fashioned cars, the ones with engines, because if you put pure water in your engine to cool it, and it froze, let's say in Michigan, the expanding water molecules would crack the block and ruin your engine. Now, I drive an electric car, so I don't have to worry about that. Most people have to put an antifreeze in there, so the antifreeze will not freeze and have the water molecules spread apart and damage the engine. What does this do? Well, it allows animals to live under the surface of frozen water without freezing. So what happens? Well, if you're a pond creature and it gets really cold, the water's going to get cold, yes, but a layer of ice is going to form on top. Now, what we're going to see in just a moment is this ice is actually fluffier than liquid water, and it acts as an insulator. It protects the liquid below it from freezing. So animals like frogs and fish can live in a pond without freezing to death, because the ice on the top acts as like a thermal blanket protecting the water below it. Another thing you need to know is water is called the universal solvent. It will dissolve more types of substances than any other liquid, and it's the main ingredient in cells. It will dissolve almost anything. The only things that it will not dissolve are things that are non-polar, such as oils, gasoline. And so those things need a different kind of solvent, a non-polar solvent. Okay. There's Kobe, who passed away recently, but I still like using his picture here. By the way, I will be asking you to tell me about some of these pictures in an essay. So what I'm going to be doing is I'm going to ask you first about this. What are these hydrogen bonds showing? Well, let's go through it and take notes on what I have to say. Now, in liquid water, when it's above 0 Celsius or 32 degrees Fahrenheit, notice the hydrogen bonds are very close together and they're constantly breaking and reforming. If you look at hydrogen bonding in a little video, you will see this. These water molecules are moving past each other, and the hydrogen bonds are constantly breaking and reforming. And this allows the water to get out of the way when you jump into it. Notice how closely packed they are together. The water is dense. When water freezes, though, the bonds become rigid and they spread out. The ice now is much less dense. Notice that there's empty gaps here, making it lighter per volume. That's what allows it to float. So I'll be asking you, why is it like this? Why does ice float? Why does liquid water get out of the way? It has to do with the fact that these hydrogen bonds are rigid and these are breaking and reforming. Now, down at the tree, remember I talked about adhesion and cohesion? Well, here's water molecules adhering to the inside of this tube. These are the water tubes inside a tree that allow water to get all the way up to the leaves. You should know that there's no pump pushing water up to the leaves in the tree. It's just the same phenomenon that moves water up your paper towel. That is, these water molecules are climbing up along the walls, bringing with them more water molecules. So because they adhere to the surface and because they adhere to each other, they pull the ones in the middle up with them. These tiny tubes are what allow the water to go all the way up 300 feet into a tree. This water bug shows surface tension. You can see how it's standing on the top of the water, indenting the water. It's because the hydrogen bonds become especially strong on the surface. And Kobe shows the evaporative properties of water. So as he sweats, or as he used to sweat, air would move across his skin, evaporating the water, cooling off the water, cooling the blood, and sending it back into his body. Acids and bases. Acids are chemicals that release hydrogen ions, which are hydrogen plus ions, actually protons, when mixed with water. Acids usually taste sour. So for example, citric acid in citrus fruits tastes sour. Bases are chemicals that release hydroxide ions when mixed with water. A good example of that is lye that you could use to dissolve a clog in your drain. Bases are usually slippery to the touch and bitter tasting. That's one of the reasons why soap tastes bitter, is because it is slightly basic. What is the pH scale? Well, you've probably heard of the number line, 0 in the middle, negative numbers to the left, and positive numbers to the right. The pH scale is similar, except the middle is not 0, it's 7. Right in the middle is 7, with 14 being at one end and 0 being at the other. Please notice the smaller numbers are acidic, the bigger numbers are basic. So don't just assume bigger numbers mean more acid. Neutral 7 is neither acid nor base. 14 is base, 0 is acid, or the most extreme acid, most extreme base. Anything on this side, anything on the pink or red side is going to be acidic. Anything on the blue side is going to be basic. The pH scale measures the acidity or basicity of a solution. The numbers range from 0, which is a strong acid, through 7, which is neutral, to 14, a strong base. So notice these different examples. Sulfuric acid you could use in lab. Stomach acid, lemon juice here at 2, grapefruit juice, tomato juice is even acidic, and urine is slightly acidic. But human blood, pure water are neutral, and then seawater slightly basic, milk of magnesia even more, ammonia even more, bleach even more, and oven cleaner or lye is extremely basic. Okay, what is a buffer? Buffers are substances that resist pH change. Now that means when you take this material and add a little bit of acid to it, it absorbs it. They accept hydrogen ions when they're in excess. So if you have a particular buffer and you add a little bit of hydrogen ion, which is acid, it will absorb that and not change pH. They donate hydrogen ions when they're depleted. So for example, if you added some hydroxide ions, it would release hydrogen ions, keeping that pH nice and stable. Your blood contains buffers to help balance the amount of acid and base in your body. So why does the acid change? Well, we'll be talking more about it later, but it has to do with the fact that when you hold your breath and build up carbon dioxide in your blood, it makes acid called carbonic acid. That carbonic acid is absorbed by the buffer, the bicarbonate buffer in your blood. Okay, that is chapter 2A. So let's go to chapter 2B. Okay, we're going to continue what we were talking about, more chemistry. This is organic chemistry. Molecules containing carbon. Okay, a molecule is when two or more atoms are connected together with a bond. This is a refresh from, or reminder of what we talked about just now. A molecule that contains carbon is called organic. Now in food, we call organic something that doesn't have pesticides in it or synthetic pesticides. It turns out organic food can still have pesticides as long as these pesticides are naturally occurring. But in chemistry, organic means that the molecule contains carbon. So it's a little different than how we use it in food. If a molecule does not contain carbon, it's called inorganic. The study of chemistry inside of living things is biochemistry. This is most of the rest of the chemistry that we'll be talking about here. Don't worry, we won't be talking too much about chemistry for the next chapter or two, and then we'll be moving on to cells and other living things. Okay, carbon is the key to organic chemistry. Since carbon is in family 4, remember I told you it was in family 4, that column of the periodic table, and it seeks to have 8 electrons total in its outer or valence shell. It will form 4 bonds and thus pick up 4 electrons. So notice here, 4 electrons are being shared here because of 2 bonds. Here, you get 2 here. For carbon, this was oxygen. Remember, carbon needs to make 4 bonds. 1, 2, 3, 4. Now, you can count each bond and the electron between them for both sides of the bond. So notice the carbon has 4 things touching it, 4 lines touching it. The hydrogens have 1. Carbon can form single bonds, which are the single lines. It can also form double lines, or double bonds, excuse me, shown by the equal sign. And then triple bonds down here, which would be the underline equal sign. That would be 6 electrons being shared, a total of 4 around each carbon. What are carbohydrates? Well, carbohydrates are a big component of our food. Carbohydrates are composed of carbon, oxygen, and hydrogen atoms. That's why they're called carbohydrates. So it's like hydrated or water-infused carbon. Simple sugars or monosaccharides are single sugar molecules. So mono means single or one, saccharide means sugar. So for example, Karo syrup is glucose, single sugar material. Double sugars or disaccharides, di means 2, are double sugars. An example of it double sugar is sucrose or table sugar. That's what you would add to coffee or iced tea to make it sweet. That has 1 part glucose and 1 part fructose. When many sugar molecules are linked together, it's called a polysaccharide or a starch. Examples of starches include many of the foods in the paper that you're writing on. Even the paper that you're writing on, if you printed out your notes, is made out of starch. That's called cellulose. We'll talk about that in just a moment. So potatoes, interestingly, made of pure sugar. That's a whole bunch of glucose molecules hooked together. Starch, pasta, all sugar. But we are going to be talking about why potatoes and pasta are better for you than is simple or disaccharides in a bit. Okay, let's first talk about how you connect together individual sugars to form a larger molecule. Well, this is called dehydration synthesis, and I will be asking you about it, so take notes. Notice that this glucose has an OH on this side, and this glucose has an OH on this side. So how do you connect them together? Well, you use an enzyme we'll be talking about in a later chapter. But when you do that, this hydrogen comes off and this OH comes off, forming water. So what is dehydration? It means take out water. So you've removed this molecule of water. What does synthesis mean? It means joining together. So this oxygen is left behind. And now we can see these 2 glucose molecules are hooked together with the oxygen in the middle because we took out water. Dehydration, take out water, synthesis, put together. So we were able to make a molecule of maltose by connecting 2 glucose molecules to each other through dehydration synthesis. We're also going to be talking about the opposite process later on that would break these apart. Please remember that term, dehydration synthesis. Take out water, join together. One familiar example of a polysaccharide is starch. Plant cells store starch for energy. This is one of the main things they store. Most of the material that we get from plants is starch. Potatoes and grains are a major source of starch in the human diet. Animals store excess sugar in the form of a polysaccharide called glycogen. And since you are an animal, you do it too. So where are you storing this glycogen starch? Well, mostly in your muscles and in your liver. So if you need sugar for exercise or between meals, your body will release the sugar by breaking up this starch and giving sugar to your body. Glycogen is similar in structure to plant starch, but it's made in animals, not plants. All right, what is cellulose? Cellulose is the most abundant organic compound on Earth. So make a note of that. I believe it's a review question and a test question. It forms cable-like fibrils in the tough walls that enclose plants. So around each plant cell, you're going to have a thing called a cell wall. What makes up that cell wall is these fibers that are made up of cellulose. And let's take a look at the structure of cellulose. Now, we can't digest it. The reason why we can't digest it has to do with these oxygen atoms here. Notice in the first one is pointed down, and the second one is pointed up, and then down, and then up, and then down. Because it's alternating like that, we can't digest it. And that is fiber. We don't digest fiber, but it does go through our digestive tract, keeping us regular, helping things move through. So if you're not passing food through as well as you'd like, you're constipated, you might take more fiber. I recommend more vegetables. If you're older, you might do this Metamucil, but either way, it stimulates your digestive tract to move things through. Now, some animals, like animals that eat plants, can break this down, but I'll show you how they do that. It's a major component of wood. So the cell walls of wood, of plants, are made out of cellulose, and that's wood. It's also known as dietary fiber, as we see in Metamucil. Okay, let's take a look at this cow. Now, it turns out the cows can eat plants, but they themselves don't do the digesting of the cellulose. What do? They keep bacteria that you see in the picture inside their stomachs. How do the grazing animals survive on a diet of cellulose? What they do is they have bacteria in their digestive tracts that secrete enzymes that break it down. So it's a form of symbiosis. And what do we mean by that? Well, the cow does the work of having the stomach and getting the chewed up plants into its stomach. The bacteria has the job of producing the enzymes. So when the enzymes break down the starch in the plants, the bacteria get some and the cow gets some, and it both works out. So these bacteria actually do the breaking down, but the cow finds the food and puts it in the stomach. So it has a really big, now cows are not fat normally, but this big round stomach has to do with the fact that you have to put all that vegetable matter together. The bacteria are there, they start to ferment and break it down, and then they share the food molecules. Okay, so let's take a look at different kinds of polysaccharides or starches. This is the starch that you would find in potatoes. Notice here, all these oxygens are pointing downward. What does that tell us? It tells us that we can digest that particular kind of starch. But the glycogen that you find in mussels, notice it's about the same in its basic structure, but here there's some branches. So more or less it's the same, except this is branched and this is straight. But again, you could consume the starch, and when you eat a steak, you're getting some starch in there, in glycogen. So you can break down these two. But look down here in the cellulose of the plants, up, down, up, down. So we can't break this down, and we don't keep bacteria in our stomachs to do it. That's why we don't get any nutrition from plant cell walls. We only get nutrition from the starch found within plants like potatoes. Okay, most macromolecules are polymers. What is a polymer? Well, a polymer is a series of individual molecules or atoms hooked together in a long chain. Polymer, poly means many, mer means unit. So in this case, we're talking about any very long chain, for example, starch. So how do we make the starch longer? How do we make the polymer longer? In the same way that I told you about dehydration synthesis. You take out hydrogen on one side and OH on the other, remove the water, and now you've added an extra piece on. And this is how starches are formed. You put an extra sugar on the end and end to end, and eventually you get a really long molecule called starch. Polymers are made by stringing together many smaller molecules called monomers. A monomer would be one thing. This is really a monomer, but it's also potentially a short polymer. Remember dehydration synthesis? This is it, dehydration synthesis. All right, now this is different because in this case, we're not making things longer, we're breaking them apart. So organisms also have to break down macromolecules or large molecules like starch. Cells do this by a process called hydrolysis or hydrolysis. Hydro means water, lysis means to break or split. So notice in this case, water's not being removed, it's being added, but it's being split apart. So if you remember, dehydration synthesis was putting hydrogen and hydroxide together to make water and removing it. In this case, you're adding water, splitting it apart, and splitting the two pieces apart. Hydrolysis, splitting water. So you split the water molecule, put one part on this side, one part on that side. Now the molecule is broken. So on the test, I'll be asking you to compare and contrast hydrolysis and dehydration synthesis. They're opposites of each other, but you need to be able to explain it for full credit. Okay, lipids. Lipids are commonly called fats, but include phospholipids and steroids. Well, this guy isn't fat, but it's possible that he built those muscles by using steroids called anabolic steroids, anabolic meaning building. So steroids, phospholipids, fats. Please add your notes, oils and waxes. So right here in your notes, add oils and waxes are also lipids. Most lipids are hydrophobic, meaning that they don't mix well with water. So you've heard that oil and water don't mix. There's a picture of that. Why is that the case? Oil is non-polar, water is polar. So because oil and water have different polarity characteristics, they don't mix together. Phospholipids compose most of the cell membrane in most organisms. So the outside of a cell is made up of this material called a phospholipid. And it's really interesting because you have a hydrophobic or oily tail down here in gray, and the hydrophilic or water-loving head, phosphate. So because it has these two pieces, the oil parts stick together and the water sticks to this yellow ball, and this forms a membrane. And it does it automatically. Take this material and mix it with water, you're automatically going to form this membrane. Most lipids, except for steroids, are composed of a fatty acid molecule that's been bonded together using a molecule of glycerol. I'll show you how that works later. Okay, this is fat. So this is what is called a triglyceride. Why is it called a triglyceride? Because we have 3 fatty acids sticking out from a molecule of glycerol. So the glycerol is in green and the fatty acids are in yellow. And here they're all together. So how did we make this? Well, if we take out water and connect these together, what do we call that? Remember, dehydration synthesis. Take the water out, connect it together. So each of these was connected by dehydration synthesis. Now, what do you notice about these 3 fatty acid chains? Well, they're different from each other, right? This one is shorter than this one. So one of the ways that fatty acids can vary, and this is a test question, so write this down, is length. This one is longer than this one. The other way that they can vary is by saturation. Now, what do we mean by saturation? Well, look right here where the bend happens. There's a double bond. Why is that happening? Well, notice that there's only CH here and CH here, where up here there are CH2s. Now, these 2 top ones, because they have CH2 in every spot, except for the beginning and end, they're called saturated because you have as much hydrogen as you can possibly fit in there. So the carbons are attached to each other left to right with the hydrogens above and below. This is a shorthand way to write it. Now, when you put a double bond in there, it doesn't leave room for those 2 hydrogens on either side. So this is called a unsaturated fatty acid because you had to remove 2 hydrogens to make this double bond. You may have also heard the term polyunsaturated fat. If there's lots of double bonds throughout, it's called polyunsaturated. Those are good because they're less likely to lead to heart disease and death. Okay, what is an unsaturated fatty acid? Well, they have less than the maximum number of hydrogens bonded to the carbons. So again, right here, carbon and 1 hydrogen each. You had to take a hydrogen off of each side, we saw up here, and to make room for that double bond. So that's unsaturated. It's not saturated with hydrogen. Saturated fatty acids have the maximum number of hydrogens bonded to carbons. That's 2 at each spot. So these are saturated because there's no double bonds, full hydrogens throughout. This is unsaturated because it's missing 2 hydrogens as a result of the double bond. So I'll be asking you, what is the difference between a saturated and unsaturated fatty acid? It has to do with these double bonds in unsaturated fatty acids. Proteins, steak, hair, amino acids. Proteins make up muscle tissue, hair, and enzymes that cause the chemical reaction inside of our body. Proteins always contain nitrogen, not found in most other organic molecules. So please note, I'll be asking what 2 kinds of biomolecules always have nitrogen, and protein is one of them. Proteins are composed of long chains of amino acids. The behavior of a protein is based on how the amino acids are arranged together. How did we put these 2 amino acids together? There we go, just like we saw before, dehydration synthesis. So now they're connected together. This is called a peptide bond because it's joining together 2 amino acids. The bonds between amino acids are called peptide bonds. That's why sometimes proteins are called polypeptides, because there's many peptide bonds joining many amino acids to each other, polypeptide. What determines a protein structure? Well, a protein shape is sensitive to the surrounding environment, which means, so I'm going to be asking you, what is it sensitive to? Well, it's the surrounding environment, but I'm going to ask you to go one step further. Unfavorable temperature and pH changes can cause a protein to unravel and lose its shape. So when I say its environment, I mean the temperature and the pH, the acidity or basicity of its environment, not just its environment. So make sure you know the environment refers to the temperature and pH. This is called denaturation or being denatured. So if you overheat a protein or change the pH too much, it's going to become denatured or change shape. So this is a picture directly from your text. These are the amino acids in a protein. This is called the primary structure. It has to do with what amino acids are hooked in what order, the primary structure. Please make a note, the primary structure is the most important thing determining what kind of protein you have. This, the chain, first, second, third, fourth. What amino acids are first, second, and third is the most important thing determining a protein's characteristics. Now, if we look at the secondary structure, look, we've taken this chain and folded it into a folded sheet, kind of like a piece of paper, or a spiral, like a spring. This is called the secondary structure because it has to do with basic shape. If we take some of these pleated sheets and some of these spiral alpha helices and put it together, we get what is called a tertiary or third level. Notice some folds, some spirals. And then if you take several of these pieces and connect them together, you get what is called a quaternary protein structure. If you want to see this, this is in your OER online text. Okay, nucleic acids, DNA and RNA. DNA is abbreviated that because it's deoxyribonucleic acid. So D and the N there, and then the A, deoxyribonucleic acid. It carries genetic information. We're going to be talking a lot about that later on. DNA contains large amounts of nitrogen structure. So remember, I'm going to ask you what biomolecules have protein in them. The first was, have nitrogen in them. Let me repeat that. What biomolecules have nitrogen in them? First one was protein. The second one is nucleic acids. Both DNA and RNA have nitrogen. So that's the question. There's a double helix formation. So helix means spiral, double because there's two spirals connected together with these bases. It's found in the nucleus of every cell in your body, except for red blood cells. We'll talk more about that later. RNA is ribonucleic acids used to carry information about the making of proteins from the nucleus to the ribosomes, where protein is formed. I'll be talking more about how that works later on. But DNA and RNA are what are called nucleic acids, and they have nitrogen structure. Okay, these are the 4 bases that you would find in DNA. Each DNA nucleotide, which is a component, is made of one of the following bases. Thymine and adenine bond together, hence the green color, and guanine and cytosine bond together, hence the brown or orange color. Adenine is A, guanine is G, thymine is T, and cytosine is C. Okay, so that is it for today. I'm glad you're watching. Be sure to work on review questions because those review questions are going to be your test questions. Those are at the end of your lecture notes. See me in lab for questions, and hope you have a great rest of your day. Bye, guys.